Welcome everyone and happy Wednesday from snowy Montana. I am Cheryl Rogers, the Director of Marketing here at Golden Helix. Our webcast today will be given by Greta Lindsay Peterson and Ashley Hintz of the Golden Helix's service and support team. Greta and Ashley will showcase how easy it can be to go from an unfiltered variant file to a report with variants of interest in VarSeq and VSReports. Exclusively, I'd like to announce that for our viewers today, we have extended our introductory pricing offer on VSReports. This does give a 25% discount on pricing as well as a 15-month extended license. So if you'd like more details, feel free to email us at info@goldenhelix.com, or you can hop on our website, and request pricing there. With that, I will go ahead and pass the mic to Greta.
Thank you, Cheryl. As Cheryl stated, my name is Greta Lindsay Peterson, and I'm the Director of Services here at Golden Helix. After getting my master's degree in mathematics and statistics, I started working at Golden Helix in 2008 as a software tester. That quickly expanded into customer support, product management, and beyond, and here we are today. If you have any questions during today's webcast, please feel free to enter them at any time in the questions pane, and we will answer as many of them as possible at the end of the presentation. If there are any questions we are not able to get to, we will get back to you with the answers as soon as possible. For those of you who might not be familiar with Golden Helix was founded in 1998. Our founders spun off from early work at GlaxoSmithKline, and GSK was a key investor in the company. Golden Helix started out creating products for genomics and bioinformatics with Helix Tree, which is now SNP and Variation Suite, or SVS, and has since added 2 products, Genome Browse and VarSeq. With these products, the company has 17 years of experience in bioinformatics and has been cited in over 900 peer-reviewed publications. Golden Helix has customers all over the globe. In fact, we have customers on all continents except Antarctica. So if you know someone who will be spending some time in Antarctica, let us know and we'd be happy to give them a call or at least a free t-shirt. Just let us know in the GoToMeeting questions pane. We'd love to be able to say we have customers or someone wearing our t-shirts all over the world, including Antarctica. With that said, we serve over 200 organizations, including top-tier research universities, clinical labs, major government institutions, and pharmaceutical companies. This leads to thousands of users who trust our software in their labs and with their research. We are very proud of the fact that when you buy a software product from Golden Helix, you are getting more than just a piece of software. We have a reputation for quality products, excellent customer service, and domain expertise. We also have the experience of developing software for over 17 years with a wide range of experts on staff, including biostatisticians, mathematicians, and software engineers. We earn the trust of our customers by reaching out and understanding our customers and their needs. In order to develop software that will work for our customers, our service department has gained industry experience by completing dozens of research projects for customers to help us understand real-world research obstacles and how to overcome them. We also research relevant topics and publish blogs and ebooks on the subject to gain knowledge and share what we have learned with the community. Our team of experts is available to provide the resources needed to help complete your analysis from training to technical support. We support our customers every step of the way and understand the ecosystem of our clients. To that end, for transparency, all of our major methods and algorithms are documented, including the math, use cases, and article citations. Our software is designed for your standard desktop or laptop without needing access to the cloud. However, if you do have access to a more powerful server or computer, our software will definitely run on that and take advantage of that as well. We will be presenting reports today that were designed specifically for this webcast. If you like what you see, we can work with you to custom design your own report. Before we get into the cancer gene panel workflow, I will briefly describe what exactly VarSeq is, our data annotation sources that support VarSeq, as well as custom reports. Then I'll briefly describe the data used for today's webcast, and then move into the first demonstration portion of today's webcast. At the end of the webcast, Ashley Hintz will describe how any workflow can be integrated into automatic pipelines for high-throughput labs. So what is VarSeq? It is a powerful commercial-grade software designed for local hardware. It has the largest and most up-to-date repository of public annotations, and the attention to details in getting the public data right that we spend for our public annotations, including left aligning and multi-allelic splitting, means that our public annotations are among the best available. We also have an advanced and powerful filtering through a rich user interface. You can export multiple outputs, including VCF, Excel, or text files. And VarSeq can also perform data transformations, including VCF and GVCF merging, intelligent handling of multi-allelic sites, breaking multinucleotide variants to allelic primitives. And we also have an expression editor for creating custom variables when what you need just isn't available by default. Since VarSeq is a filtering and annotation program, it is only as good as the annotation sources supporting it. Because of this, we spend countless hours finding the best public data sources to curate and format to our data specifications. But we don't stop there. We verify that the data is accurate and represented in a way that can be used for annotating variants. When we find problems, we make sure to go back to the source to fix the issues we find. If that is not possible, we will fix the data ourselves before making it available on our public repository. Some of our data sources are updated monthly and others less often. We strive to make the updates available as soon as possible after going through our thorough QC process. Because some versions are updated so often, and we know that our users need to create a set workflow with set sources for CLIA validation, we do not automatically change the sources used for your workflow. You lock down your annotations until you decide you want to update. We also have 2 additional sources available at this time that can be added on to a VarSeq license. These are OncoMD as a source for curated drug-targeted mutations for cancer and supporting clinical trials and functional evidence, and OMIM genes, phenotypes, and variants. Finally, our custom reports, or VSReports, does come with 2 report templates with the license, one for hereditary workflows and one for cancer gene panels. However, most of our users find that these reports need customization to meet their needs. All of our report template and framework files are available to the user to customize. However, if you find that you do not have the resources in-house that can make the customizations, we can help you with those customizations. Small changes can be included in the VSReports package, while more extensive changes would be possible for a small fee. On the left here, you see the default PDF version of the cancer gene panel report, and on the right, you can see the customized version. We'll take a look at the version on the right in more detail here in a few minutes. Finally, the sample data that we're going to be using for the cancer gene panels comes from the Illumina TrueSight myeloid sequencing panel. It consists of 3 replicates of NA12877 with different percentages of Horizon DX known somatic mutations mixed in. This data from BaseSpace comes with everything we need for the cancer gene panel, the BAM and VCF files, as well as the targeted regions in a bed file. This is also a very high coverage panel where the average read depth for all 3 replicates is over 4,000 reads. All right, let's go ahead and move into our demonstration portion. So as you can see, we have the VarSeq window open here. And within the window itself, we have these different regions. We can call these regions a VarSeq view or a tab, whatever is more comfortable to you. I personally prefer to call them views. So here we have a filter view, a table view, as well as a detail view. Now the first thing that we want to do is we want to look at whether or not our samples passed QC, whether there is good coverage for the targeted regions. If there wasn't good coverage, we don't really need to bother looking further, and we need to rerun the samples. So I'm going to open up the sample table here. And if I scroll over, I can see that I have the 3 samples at 10%, 25%, and 50% of Horizon DX. We can see that we've got the BAMs linked. Because we have the BAMs linked, we were able to compute coverage statistics for the targeted regions. And as you can see, the sample mean depth over all of those targeted regions are very good. And we can get a little bit more in depth. We can look at the percent covered at 500x. And we can see the 50% mix has a 83% of all regions were covered at 500x. So these three samples we will consider all pass QC, and we can move on with our analysis. I can see that information up here on the right as well. Again, I see three samples, and initially, unfiltered, we had 774 variants. Because this is a targeted panel, we have just a small number of variants coming in. We can work with whole exomes and whole genomes as well. And Ashley later, when she presents her demonstration, will be working with a much larger initial variant set. Now, the workflow that I did for today is based on the shipped template for cancer gene panels. However, I have made some modifications. The first change that I've made is I've added this filter up here at the top, indicating whether or not a variant is within the targeted regions for the panel. And as I can see, if I expand this, 11 variants actually fell outside of those targeted regions. So I'm only keeping the variants within my targeted regions. The next group of cards are actually, our filter cards are actually part of the default template. The next one is whether or not each variant passed the variant caller's QC. These are classifications made by the variant caller, and I only want to keep those that are marked as pass. So it does drop my number of variants a little bit. The next card here is the read depth for each variant. Now I know that all of my regions for my samples are covered at a very high depth, and so 1500 might be too low. If I look at the histogram for read depth and I scroll down, I can see, yes, I do have very high coverage. So perhaps I would want to change my threshold for keeping variants to 2500. So I'm just going to quickly modify this. And I see that I moved from 25 variants down to 21 variants. The next filter card here is for the alternate allele frequency. We want to make sure that this is greater than 1% to capture true somatic mutations. The next card is whether or not a variant is present within COSMIC, and I do want to keep those that are known COSMIC mutations. And finally, this last filter card is also not part of the default cancer gene panel template, but I have added it. This is the whether or not the variant is in has a clinically relevant has an effect in the clinically relevant transcript from RefSeq genes, the loss of function or a missense variant. So I only want to keep those that are within exon regions that have a loss of function or missense classification. With that, I get down to 21 variants. You can see here in these colored columns that I have marked a few variants, and these are flags that I have set myself. The first column is for primary findings, and the second column is for secondary findings. And as you see, I have only one variant marked as a primary finding. If we scroll over to the right, we'll see why I chose that as my primary finding. First of all, out of all the genes of my 21 variants that passed my filter, this variant is in the BRAF gene, which is a known cancer hotspot. And if I continue scrolling over, we do have OMIM information for that gene, and it is an OMIM variant, but we also have OncoMD drug targeting mutation information for this particular gene. So there is, we can take an action with this variant based on these drugs that are indicated by OncoMD. We also have clinical, if I scroll back over here, we do have clinical trial information available for this variant as well. So this would be our most interesting finding, or primary finding. I have marked 5 other variants as a secondary finding based on other criteria. Now I'm going to go ahead and I'm going to jump ahead to the end where we look at our generated report. You saw this earlier when we were looking in the PowerPoint presentation, and this is our HTML view. We can see we've got the lab information as well as the provider information, patient and sample information, and two things I want to point out are the panel coverage and the average read depth. Now this is very important information, but you do not really want to have to calculate this by hand. Fortunately, since we have computed coverage statistics, we can do this for you. So this particular sample had 86% of the panel covered with an average read depth of over 5,000. Next, we see that there is a positive mutation for this sample, and we can get a table of all the genes for this particular panel, and we can see the variants that we've marked as either primary or secondary findings within this table. Next, we have a list of all of the primary findings. So again, we just had the BRAF gene, the variant within that, and we get an interpretation summary. This is information that I manually put in. Because this is a cancer gene panel, we really care about OncoMD drug summary information as well as trial summary information. So I have a table of all of the drugs from this particular variant from the OncoMD table, and with the drug name, the generic name, and the response rate. So I can use this to make decisions or recommendations on how to proceed for this sample. Finally, I have an OncoMD trial summary. Now again, because BRAF is such a well-known cancer gene, there are over 60 clinical trials available. For this particular report, I chose to just include the first 10. We can get down into the details by having individual variant interpretations for all of the variants that I included in my report for the primary finding as well as for the incidental findings. And we automatically generate references based on the information pulled from OMIM. Finally, we have additional information that I included based on the test, the panel, and as well as the indication and the background information from that panel. And I pulled this directly from the Illumina information. And as we can see at the bottom, this is a draft report. If I want to go ahead and say, I like all this information, this is good, I approve this variant, approve this report, I can add that information in the report view. A lot of this information I've filled in about the sample, the doctor, and the sample information. Again, I have chosen the interpretation summary and the recommendations. I could change the report from positive to negative, or the result from positive to negative here. But I've selected my primary findings as well as my secondary findings here in this table. At the bottom of this report view is a box for signing this report. If I check this box, it will add my name, email address, or login information for VarSeq, as well as the date and time stamp. By doing this, it will generate information at the bottom of this report indicating that I have approved this report. Now, you don't want to send HTML information around unless HTML is the format that your EMS requires. So what you really want to do is save this as a PDF. So I'm going to go ahead and save this to my documents folder, or you can save it wherever you like. And it will render a PDF. And let me bring my PDF reader over here and make it so that you can see the whole page. And we can see that we've got all that information generated in our PDF report in a very readable format. If some of this information is too long, you have the control to delete some of the paragraphs that are maybe not necessary for your study. Because we autofill this in, it makes it easier to remove information than having to manually add information. But again, all of this is completely customizable. If you wanted your report to look different, to have different sections, to have different headers, we can very easily work with you to make that happen. All right, so with that, I will go ahead and switch over to Ashley. All right, thanks, Greta. Okay, as was mentioned, I am field application scientist for Golden Helix, and my background is actually in phylogenetics. The demonstration I'll be walking you through is for a trio analysis, and we're actually going to go through 6 different workflows, or filter chains, covering de novo, dominant heterozygous, compound heterozygous, recessive homozygous, X-linked diseases, as well as known rare pathogenic. We'll start out by looking at the different filter chains and looking at the existing template available in VarSeq. I'll walk you through the data tables and annotation sources. We'll examine any interesting findings as well as look at those in Genome Browse. We'll look at the various filters applied, and then we'll save the workflow so it can be repeated in a different project or for a different family, as well as look at VSReports. To familiarize you with the family we'll be working with, it is from the International HapMap project from the Nigeria population. The BAM files actually came from 1000 Genomes Phase 3 Illumina Exome. We've injected a variant into the BAM file of the proband and then used SAM tools for variant calling. With that, let's jump into VarSeq. Okay. As you can see, the view at the top is slightly different from what Greta was showing. I have 6 different workflows. And this is accomplished by using the logic operator inside the filter view. So when I right-click in that view, I have the option of an AND or an OR logic. The OR logic means that I can put each one of these workflows next to each other. The AND logic is what is occurring inside each one of those workflows, stacking those filter cards on top of each other. And when you set up the workflow that way, I can actually get the intersection between each one of these workflows in my 2500 variants here at the bottom. Now for today, I'll take you through the setup for each one of these workflows, and we'll talk specifically in detail about the de novo workflow. Each one of these different workflows starts out similarly with QC operations, including read depth and genotype qualities. In this case, this exome is 3x coverage, so these filters are set much lower, as we saw with the cancer gene panel, read depth of 10 and genotype quality of 20. After that, we are looking for a rare variant, so we're using the exome variant frequency track from NHLBI, setting it at 1% and looking at variants that either fall less than 1% or perhaps missing from that data source. And you can see that filter, those sets of filters are across the entire set of workflows. The next filter card is looking at the effect of the variant, either loss of function or missense, and this is coming from the RefSeq genes annotation source, which is again repeated in the other workflows. The next filter card is specific to the de novo candidate workflow. It is using a computation available in VarSeq for Mendelian error, and as you can see, we only have the de novo alleles highlighted. So if I click on those 68 variants, variant table, so I can just continue to work with those 68. Another computation available in VarSeq, if we flip to a different workflow here, under compound heterozygous, is for detecting compound heterozygous variants. In this case, it's either a true or false. It does contain a compound het or not. And of course, we have the true indicated here in green. So flipping back to our de novo workflow, the next filter card I've added to the template is looking at functional prediction algorithms. In this case, this annotation source goes through 5 different functional prediction algorithms and then tabulates the results between those 5 algorithms. And I've indicated here 3 or more that consider the variant damaging, as again highlighted in green. And that takes me to 17 variants. And I can narrow that down a little bit more by using the FORank algorithm available in VarSeq.
This is based off of the Fever algorithm. It goes through the GO and HPO biomedical ontology databases matching my phenotypes of interest, in this case neurodevelopmental delay and microcephaly, and connecting them with genes that display those phenotypes. So if I click, actually let me scroll to that data field in my table here. And if I click on that information, it will give me a histogram. In this case, I do have a large number of variants right in the middle, but let's say I'm interested in looking at anything over 60, so in the 60th percentile or higher, and that's the filter I've set here. So in this case, I just have 5 variants. Whoops, let me click that again. So now I just have 5 variants that I can continue to investigate further. And to do that, I'm actually going to use Genome Browse, because as I mentioned earlier, we have the BAM files available for each one of these samples. And I've imported them here into Genome Browse. So we'll start at the bottom of our list here and look at our first variant, which looks relatively good. I can actually click on the BAM file track here and get the information about it. So let's look at our next one. This one is actually in a highly variable region of the genome in the HLA genes. So that might not be the best variant, but we'll continue to look. This variant as well is also found in the HLA region. This particular variant, if I click on the BAM file information again, I can see 80% of the reads are for the G reference allele and just 16% are for the A, which is a possibility, but let's check out our last variant here. And that one looks pretty much to be a 50-50 heterozygous split between the reference A and the alternate G. So that might be the best variant of interest. I can also see from the ClinVar information and my OMIM information that these are confirmed variants, both of which at the top are pathogenic. And that's how I flagged my variants here at the front. My primary findings are indicated in red for those first 2 variants confirmed in ClinVar. The other 3 I've indicated as secondary findings. So from here, I can put all of this information into a VarSeq report for my trio analysis. Again, all of this information is entered manually, except for all of the variant information at the bottom, which we are pulling directly from the OMIM information. And so when I click play and generate the report, I have all that information filled in. Again, I have my average read depth filled in as well because coverage statistics were calculated, and I can see it's only at 3x. Now for the family trio information, I've also included the mother and father with their sample information for reference for the clinician in case either of those information need to be rerun. And we've indicated here the top 2 variants that are pathogenic and all of their information. And at the bottom, in this case, I have the affected genes. In this case, since HLA had 2 variants, I only have 4 genes listed. And then, of course, we're pulling all of the reference information from the annotation sources, so you don't have to manually fill that in, and the additional information about the exome analyzed. And of course, this is all exportable and be able to save it in a PDF, as you see here. So I can pass it on to somebody else in the lab, potentially. Okay, so since I've adjusted this particular template a little bit for my own use case, perhaps I want to use it in the future on a different family trio. In that case, I would need to save this project as a template. Maybe. And once you save it as a template, you'll be able to go back and use it on other projects. And that is what you would need to do if you wanted to use VS Pipeline. for a batch analysis. Okay, so the steps here indicated in red are basically the demonstration that I just showed you. We've started a project, we've defined our workflow, and then we've saved the project template. From there, the steps in blue are the ones that can be accomplished in VS Pipeline. You can start a new project with that template, interpret each one of those samples with the designated annotation sources in the project, and export the results, such as in an Excel or a VCF or a text file. And each one of those projects generated in VS Pipeline can also be visualized in VarSeq Graphical User Interface. So the deliverables in this case are going to be the exported VarSeq project as well as any Excel and VCF files you've asked it to generate. Excuse me, I was coughing. And when you deploy the project template, it will also support validation for CAP or CLIA, and it also logs all the steps along the way, so you can see who and what has been done at each step. I'm sorry, who has generated and what has been done at each step. And finally, in this command line interface, you can integrate it as part of the pipeline in your lab that also generates your BAM and VCF files. With that, I will pass it back to Cheryl, and then we'll answer some questions.
Thank you, Ashley. If you haven't had a chance to enter any questions that you have, please go ahead and do so in the questions pane. As always, I will have the recording and slides up on our website more than likely today, but if not tomorrow, and I'll just shoot those to you in an email, so look for those. And as I mentioned at the beginning of the webcast, we have extended that 25% discount offer on VS Reports for our webcast attendees today, so if you're interested, you can get in touch with us at info@goldenhelix.com or jump on our website. So with that, we will go ahead and answer some questions. First question, are annotation sources included in VarSeq for free?
Cheryl, I'll go ahead and answer that one. All of our public annotation sources are most definitely included in VarSeq for free. However, we do have two non-public annotation sources that we do have an additional charge for unless it's purchased as part of a bundle. And those two are OncoMD and OMIM. OMIM comes with by default with the report with VS Reports. If you wanted to add it on, you can talk to us about that without having reports. But it is what we call a secure annotation because we do not get access to those for free. So it is not a truly public annotation source. Anything that is available for free for commercial users, we then pass that on for free. We don't charge for free annotation sources. I hope that answers that question.
How long does it take to get from a VCF file to a final report?
I'll go ahead and answer that one. To get from a VCF file to a final report can take anywhere from a couple seconds to a couple minutes, such as running the gene panel, which has a much smaller set of variants as compared to a whole genome or exome. It may take a couple minutes for the program to finish calculating all of the annotations as well as the computations before that report can be generated at the click of a button.
Let's see, do you charge per sample or per report generated?
No, we do not. You will pay for your subscription of VarSeq as well as VS Reports, and you can generate as many reports and analyze as many samples as your research or your lab requires.
Okay, thank you. Let's see here. If you don't have parents, is there any way to filter for potential compound hets in a single proband by simply requiring that a gene have at least 2 variants?
Cheryl, I'll answer that one. Right now in VarSeq, we do not have the ability to do that. However, I'm really excited to announce to you, those of you that are listening in, that in our very next release of VarSeq, we will have an option that will basically do accounts per gene of all the different types of variants. So it'll give you a total number of variants per gene, as well as the number of heterozygous variants per gene, homozygous variants, and so on for a single sample. So it's basically compound het without any parent information. And I think that will answer a lot of needs that a lot of our customers have expressed. And again, with the responsiveness, with listening to our customers, we heard that this was a must-have, because not all the time do you want to have the idea of the potential compound het without having the parent information. We heard your requests, and so we are excited to include that in our software in the next release.
Thank you, Greta. Next question. For the report, is the pooling of information from OMIM automated in VSReports, or is that a copy, paste, or custom script?
I'll take that one as well, Cheryl. Feels like I'm playing Jeopardy here. I'll take this one for the win. It's totally automated. So all you have to do is annotate your data against OMIM, and then our reports are automatically pulling that information in. If you want to pull in other information from other annotation sources that you have already annotated your data against, we can also add that to a custom report, and so it would do that automatically. For obvious reasons, we can't automatically make an interpretation or a recommendation, so that those will never be automatically filled in unless you had a really good annotation source where you already have that information that you've generated yourself.
Alrighty, thank you, Greta. I'm gonna punt the next one to Ashley so she can get some more points on the board here. Ashley, can I add my proprietary annotation databases in as well?
Yes, you can. If your institution or laboratory has in-house annotation sources that you would like to use for annotating and filtering, you certainly have the option of converting those in VarSeq from standard bioinformatics formats such as a bed file, text, VCF, into an annotation source to use locally.
Thank you. Next question, I'll leave this one up for grabs. How are deduplicated, realigned, recalibrated SNP-F, SNP-SIFT, VCF files be uploaded to VarSeq?
All right, I'll take this one, Cheryl. I had my finger on the clicker. So as long as your VCF files are proper VCF spec formatted files, it doesn't really matter how they are generated. They can just be imported into VarSeq using our standard import wizard. So there are, again, with SNP-F or annotated files, they're a standard VCF file. So all that information, all that extra annotation that was done by SNP-F can be pulled in as well.
Okay, next question. Can we enter symptoms of the patient to facilitate data analysis?
I'll go ahead and take that one. So in the case of the family trio, using FORank allows you to enter in some of those phenotypes or symptoms that the patient is suffering so that you can identify genes in the GEO and HPO biomedical ontology that have already been indicated as correlated with that particular disease phenotype or symptom.
Thank you, Ashley. Last question I've got on the board at this point. Using the coverage computation tool, how do you suggest incorporating coverage adequacy data into a report?
Cheryl, I'll grab this one. So as you can see, our report, our basic default report templates already look to see if coverage statistics were computed, and if they are computed, they will look at the sample level overall coverage as well as percent coverage, percent of the panel targeted, and add that automatically. If there's other information from that coverage statistics that you want to have pulled in automatically, again, we can certainly do that through working with you to design your own custom report. But we already essentially for free with the basic report, pull in those two coverage statistics for the particular sample.
Thank you, Greta. We've got one last question here. Do you have license versions which are different for academic or commercial users?
So the license itself is not different. I believe this is more of an account manager question. The pricing, I believe, is different, but all versions of our software are VarSeq. The base version give you access to the same features. Then our add-on features, again, whether you're academic or commercial, you need to add on VSReports or VS Pipeline, OMIM, OncoMD. We treat academic and commercial the same. It's just that the price point might be different.
I can chime in on that one as well, Greta. The price structuring is a little bit different for academic versus commercial companies. And then we do have a lot of variations in our licenses for what your specific lab needs are, whether you're a small lab or a large lab, we can scale with you. With that, I'm going to go ahead and wrap it up. And like I said, I'll have the recording and slides out to everyone. I very much appreciate the time today from Ashley and Greta and everyone out there in the audience, and we will see you again soon.
Thank you, Cheryl.